And thank you again for this fantastic meeting. So I'm going to try to give a very brief update. A lot of this is triggered by all the excitement around the otoferlin clinical trials. And certainly we're here in Japan. And Japan is really further along than any other nation in the world in terms of molecular diagnosis for genetic hearing disorders. They've accumulated an incredibly large database. So the biggest other group in Iowa has about 1,000 patients sequenced. And I think the last I heard, they had done 20,000 patients at this point. That's correct? That's the correct number, right? Which is stunning. So I really think Japan is going to become an epicenter for gene therapy research. So we as surgeons are not going to go out of business. That's my take-home message. Just in brief, gene therapy is a sort of broad term for using either both viruses and non-viral vesicles to transfer a nucleic acid, either DNA or RNA, into a cell and thereby changing the behavior of a cell. So you can use this to augment the behavior. You can put in genes that weren't there before. You can replace deficient genes, or you can even edit or remove genes you don't want to be there. So it's really just a technique. And at this point, there have been three groups of human studies using very different techniques. So this is here and now, and has been here and now for about 10 years or so. So this is my study, the CGF-166 study, where 18 patients were injected with an adenovirus carrying a transcription factor to regenerate hair cells. We had 2 responders based on our criteria and no adverse events. Our big problem was we probably lacked diagnostic clarity on whether the severe to profound hearing loss patients we had actually had hair cell loss or some other disorder in the cochlea. So the diagnosis is so important. First presented at the European Society for Gene Therapy in 2023. There are 5 different groups using different AAV-based delivery methodologies to treat otoferlin. This is a popular target because in otoferlin we know the hair cells are there and we have a genetic diagnostic test. Some really good responders, some complete non-responders, and some in between. But at this point, about 20 patients treated without any adverse events. Also, the BADGE trial, this is an Australian study where a plasmid, so just essentially naked DNA containing neurotrophin genes, is injected into the inner ear prior to cochlear implantation. The 1-year outcomes were just presented in Kuala Lumpur. 16 patients treated with clear electrophysiologic benefit to neurotrophin therapy and cochlear implantation. Compared to controls. Again, no adverse events. So this is something that's here and now. So I'm going to focus the rest of the talk on just the genetic disorders. We heard a little bit about this already. Really, really important is that the cost of sequencing is rapidly coming down, and it is at this point cheaper than getting an MRI, at least in the US. So this is something that we should be doing for all our patients. There's still a lot of challenges, and we can't treat everything. So this little word cloud, the size of the letters corresponds to how common the disorder is. We have several major problems. A lot of the human deafness disorders have their human hearing develops during the 2nd trimester. And for the genes that are active and prevent development of the inner ear properly, you lose hearing in utero. So to fix this, you would need in utero gene therapy, because once the child is born, the end organ, the target organ, has degenerated, and all that we can do at that point is a cochlear implant. Size of the target gene is important. I won't get into that. And dominant disorders, you need to treat with gene editing, which is not quite ready for prime time yet, but we're getting close. If we put all of this together, our ideal targets are recessive genes, ideally with a little bit later onset, so that we can find the patients. Very important also, you want a human population there. If the disease is too rare, you're never going to get a big drug company to finance the very expensive process of developing a gene therapy. So if we put all of this together, we have in light blue the smaller genes that can be fit into a smaller AAV vector. In pink, you have the bigger genes that need two separate AAV vectors or a larger virus like a lentivirus. You have the incidences on the right column, and the stars in the left column are where my lab and research group has preclinical data. For TMPRSS3, we're just going back to the German regulators with that, and we should have a clinical trial hopefully next year sometime. Notice also under locus, a lot of these genes come in dominant and recessive flavors. So really what we need to be targeting for now, at least in the very early stages, is the recessive types of the genes. So again, sequencing and a correct molecular diagnosis is of the utmost importance. I'm going to spend the rest of my time talking about myosin 7, which is something I'm really pleased to announce MED-EL will be developing as a gene therapy for its patients. So the Usher's disorders are a series of genetic disorders where you have both hearing and vision loss and variable vestibular impairment. One of the most common ones, Usher 1B, is caused by a defect in the myosin 7 gene. And in the full syndrome, you have a child that's born profoundly deaf with complete vestibular areflexia and onset of vision loss at around age 7 in most cases. So if you're a homozygous mutant, your tip links don't work and the hair cells can't depolarize. This is an available mouse model for that, and you see the mouse circles. Yeah, and it's non-affected. Kin can climb up on the cage, and this was actually described by my original mentor, Bob Rubin, in 1967. So I'm really happy to be working on this. And we can identify both wild types and recessive homozygous mutants and heterozygous mice by PCR. So we have an assay to check them. So this shows the spectrum of mutations in human Usher syndrome. And depending on where the mutation is, you get either the full deaf-blindness syndrome, you can get recessive hearing loss alone, or you can get dominant hearing loss alone, depending on where the mutation is in the gene. So again, molecular diagnosis, super duper important. In the mouse, what's interesting, if you have a mutant, you lose hearing sometime between day 14 and day 30 of age. Wild-type background have no hearing loss. And the heterozygous, so if you have one broken copy of the gene, you have no vestibular problems, but you have a progressive hearing loss over 6 months. So the gene can act like a recessive or a dominant gene depending on how many copies you have. So we've studied this a couple of different ways. So if in the mutant condition where you have two mutant genes, the hearing is not rescuable, and these children are essentially born deaf. But what's really interesting about this is that the vestibular hair cells persist, and we know this from human temporal bone specimens, and the vestibular disorder, so the circling in case of the mice, can be rescued up to, we've looked in up to 6-month-old mice, so a quarter of a mouse lifespan. So what we are actually preparing to do, I'm going to talk about this first, is there's a way actually of tracking balance in the mice. If you look at an unaffected mouse, it's sort of on a little force pad and it's wandering around its cage. The shaker mice that are homozygous mutants turn in one corner, And now this is a shaker mouse that's been treated in one ear with a virus that restores normal myosin 7. And they're sort of in between. They can walk around, but they don't stay stuck in one corner. And we will be developing a clinical trial for co-administration of this virus with myosin 7 for a vestibular disorder along with a cochlear implant. So we'll fix the hearing with a cochlear implant and fix the balance problem with a virus. Now, for the sort of dominant disorder, we've looked at this a couple of ways. If we put a huge excess of normal myosin 7 gene into these animals, we can rescue the hearing, but we can also rescue it through editing, which sort of broadens the application to a lot more different genes. So in editing, what we do, everybody's heard of CRISPR. CRISPR, Cas9 is a bacterial protein it's a bacterial defense against virus infection, actually. And it's been co-opted and used to cut DNA and repair DNA. And what you need is a segment of RNA called a guide RNA that brings the Cas9 protein to the correct point in a gene. And then what happens is the Cas9 will cut the DNA. In cells that turn over, that divide, you can actually get a complete repair. In cells that don't divide, like in the cochlea, what you essentially can do is use the guide RNA to make cuts and destroy the gene you don't want to be there, or a copy of the gene. So basically, you can knock out the one deficient copy of the gene. So we take our Cas9 and our guide RNA, and we actually package them into little vesicles called exosomes, which are made by all cells. And then we can use that to dose the inner ear. So this is, I just want you to take a look at this down here. These are EVs from mesenchymal stem cells that have Cas9 and a fluorescent protein in them, and we can get them into all of the hair cells, into the spiral ganglion cells, and so we're able to deliver the editing molecules to our target cells. This shows the outcomes. This is a treated ear in ABR in a mouse showing thresholds down to here. This is the contralateral untreated ear showing a fairly significant hearing loss. So we're basically able to rescue the hearing using editing in the dominant version of myosin 7. It's about a 10-year trip from these proof of concept experiments to an approved drug. So we have a little ways to go, but we're really moving forward. And sort of to finish things, I just want to show you this. This is sort of a concept that Atanaja Warnecke from Hanover has developed, and she sort of thinks of hearing in terms of different zones. So we have sort of a ski slope hearing loss. In the worst hearing portion, probably our target tissue is so degenerated we will not be able to regenerate it or rescue it with gene therapy. In the middle, in the pink zone, you have a regenerative bridge, which is amenable to therapeutic correction. And then in the normal hearing section, and we've heard about this yesterday too for the progressive hearing losses, we can probably rescue hearing and prevent progression. So I think what the future holds for us is a combination of molecular therapy and cochlear implants, which I think will be very exciting. This is my research group. And again, I'd like to thank all my collaborators, the gene therapy labs in the Department of Hematology in Hanover, and certainly MED-EL for all their support and foresight. Thank you.
Thank you, Hinrich. Sorry, is there any question or comments? Sorry. Oh, okay. Please. Use microphone.
Excellent presentation, Hinrich, as usual. Can you give us an update? I missed some data about the otoferlin gene therapy, which I think there's already some studies in humans.
Yeah, so we had a meeting an inner ear biology that Hubert Löwenheim and I co-organized, where we invited all the different groups to speak and show their data. At that point, I think 22 patients had been officially treated with available outcomes data. And they are using different vectors, different promoters, different delivery points. And I think the take-home message is the inner ear is open for business. You can treat it lots of different ways without ruining residual function, which ultimately, that lesson comes from hearing preservation surgery. You know, as soon as we were able to put a big old electrode in the cochlea and save hearing, it was clear that you can inject things. And I think we now have data from multiple different studies looking at different outcomes that putting a virus in or foreign DNA in does not result in complete cochlear fibrosis. So, for example, my patients, they were followed for 5 years with serial yearly MRI scan to make sure that we did not induce any inflammation and fibrosis. So at this point, we know quite a bit. Some of the children that were in the otoferlin trial have done spectacularly well and have, you know, some have not had any response at all, and it's not clear what the differences are. And all the groups have had both good responders and non-responders. So I think we have a lot more to learn.
Mm-hmm. Sorry.
I have one question.
Yeah,
Thank you very much.
Over there.
yeah.
No.
No.
Professor Kawasaki.
Okay, thank you very much. Thank you very much. Excellent talk. I have only a question about your studies about Mayo 7. You said for the vestibular system, you use something for the Mayo, and for the cochlear only a cochlear implant. Is this right? Or it's not the moment now to make a vestibular cochlear implant?
Well, I think I still think if you can get a rescue without lots of extra hardware, that's a good thing. What I think is really interesting is that the pathology is markedly different in the vestibular system and in the auditory system. So the auditory hair cells in multiple different mouse models degenerate. The vestibular hair cells in multiple different mouse models and in human temporal bone pathology are maintained. So maybe even as we get on with this, if we have patients that already have an implant in, we will be able to treat them afterwards and at least rescue their bilateral vestibulopathy. So that, you know, the 3 hits, we can fix the hearing in the patients with an implant, but being blind and having bilateral vestibular hypofunction is a huge disability.
So for myosin 7A, target will be the usher.
Initially usher with the dominant progressive ones, which are common in Japan, is the
it's a
second.
dominant progressive hearing also is very common in Japan. So if gene therapy can stop progression, it's a very good story. Yeah. Sorry. Yeah.
Thanks for a great talk as usual, Hinrich. So I have a question on the durability. So we know the cochlea is open for business, but for how long is it open for business?
So in hematology, there are non-human primate studies where they have gene replacement. So these are studies targeted ultimately for sickle cell anemia and the beta thalassemias. In monkeys that are long-lived, they've had gene expression of foreign genes for hematologic disorders for over 9 years. So at least nine years, and there's no reason to think that there's going to be a downregulation.
Any other? Yeah.
Great, Hinrich, for these presentations.
It's always a concern with this gene therapy if they end up with the inflammation, fibrosis, and then we lose the cochlea and not able to do a cochlear implant. And I heard few people nowadays with having a hard time with the insurance companies to fund bilateral cochlear implants because there is a hope that gene therapy will be helpful for the other side. But I'm now hearing that the idea now is just using it even with patients with the cochlear implant. So this is a good news that we could argue with anyone who said wait for the other side, that even gene therapy can be used with cases with the already implanted with cochlear implant.
Correct, and I think that's the message, especially to parents, but I think it's critical that we have structure preservation, even if there's no hearing. So even if there's no hearing, the insertion needs to be atraumatic.